Hi, this is Dr. Kat from Central New Mexico Community College. This is our digestive system video N, the second to last video on the lengthy digestive system. We will focus on the digestion and the absorption of lipids in this video, which is a pretty complex process. Unlike amino acids and carbohydrates, even nucleic acids, lipids do not dissolve well in a watery solution such as our chyme. They're hydrophobic, and therefore it's a much more complex process to break up our lipids into smaller and smaller components to where they ultimately can actually be absorbed. So let's take a look at some of the major steps. So whatever lipids that we ingest, which we're going to focus on the triglycerides primarily. Yes, they get tackled a little bit by the lipase produced by the tongue in the mouth, and the stomach also produces a lipase, but really it's very difficult for those enzymes to really break up these triglycerides. They can really just work on the very outside of the blob of lipid, really. So we end up with these lipid droplets in the stomach eventually, really mostly due to the churning actions of our stomach. It's not until we get to the small intestine that these lipid droplets that we formed now are going to be dealt with. And one of the first processes that we will see happening is something called emulsification. Essentially, the bile salts in the bile that is secreted in the small intestine are going to almost act like a detergent that breaks up a big blob of fat into smaller and smaller droplets. There's also some pancreatic lipase, and eventually, and we'll look at this in more detail here in just a moment, we end up with these very, 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 very small little fat droplets, which we call micelles. These micelles are still not ready to be absorbed, and eventually they need to be repackaged inside of the epithelial cells to ultimately form chylomicrons. Now, it says here that these chylomicrons are going to be transported in the blood, but initially they actually enter the lymphatic system. And ultimately, we see the breakdown products of our fat entering into the bloodstream. So these chylomicrons initially enter into our lymphatic system, more specifically into the lacteals that we see in the villi of the small intestine. Bile is needed in order for us to digest the fats we ingest. And just as a quick reminder, this is a slide we have looked at before. Remember, bile is a alkaline solution, so it has a slightly higher pH, which contains bile salts. They are going to play an important role with the emulsification process. We also find the pigment, particularly bilirubin. Remember, bilirubin is ultimately produced from the breakdown of hemoglobin. And we also see some other lipids such as phospholipids, cholesterol, triglycerides. So we see that basically bile is a lipid-rich substance that helps with the breakdown of other lipids. And let's not forget some electrolytes. So those are some of the main components that you need to be aware of. Be sure you definitely remember that it's made up of bile salts, bilirubin, and there's even some cholesterol present. So these components of the bile, particularly the bile salts and the phospholipids, they're going to allow for that emulsification process to occur that ultimately leads to micelles, so that we can absorb our fat and even our cholesterol, and it helps. They also help solubilize the cholesterol. So remember, once again, lipids are hydrophobic, and so they cannot easily be absorbed in their original condition. We're going to have to transform them. Now, if we go back to the chemical structure of the neutral fats, also called the triglycerides, remember that they're made up of a glycerol backbone with fatty acids dangling off of them. And with the help of lipase, these triglycerides can eventually be broken down into their individual components to the point that we ultimately might end up with some fatty acids. But this is a pretty slow process. These lipases can only end up working on the surface of these flat fat globules that float around in our chyme. So fat can't dissolve in a watery solution. So what happens to that fat that we eat? Well, it's barely gets touched on by lipases in the mouth and even in the stomach. So if we were to think of the chyme in the stomach just sitting there for a while, like the dishwater in your sink, then you could visualize that the fatty layer will reach the surface of the chyme. Similar principle to when you do dishes after a fatty meal, you're going to have this fatty layer reaching the surface of your dishwater, right? It can't dissolve in the water. What do? You add detergent to your dishwater to break up that fat layer into smaller and smaller and smaller and smaller little droplets, fatty droplets. And that's exactly what we're going to see happening during the process of emulsification. But let's go back to that fatty layer on the top of our chyme. Well, that gets broken up somewhat with the help of the stomach's churning, obviously, so that we might end up with some big fat globules. And the big fat globules that ultimately make it into the small intestine can now go through the process of emulsification, to where bile salts and phospholipids can break up the fat globule into what we can call emulsion droplets that are quite small. They're only about a micrometer in diameter. And so due to the polar and nonpolar properties of the phospholipids in particular, we can see this happening, the formation of these little, I'll call them emulsion droplets, or they call them fat droplets here. Once we have these emulsion droplets that are coated with the bile salts and the phospholipids, we have small enough structures to where the lipases can now go to work more effectively. We have small enough little droplets to where the lipases can actually digest the triglycerides inside of the emulsion droplets. And remember, a triglyceride is made up of fatty acids and a glycerol, and we can take off one fatty acid at a time to ultimately end up with monoglycerides and fatty acids. So that's exactly what happens. And this results in the formation of even smaller little droplets, which we call micelles. So micelles are literally monoglycerides and their fatty acids coated still with those bile salts and phospholipids. And there's some other components still added on to. Remember that the bile has also some cholesterol and electrolytes, etc. Now these micelles are so small, remember we expressed them in just nanometers, to where they very easily can sneak in between the microvilli here, or the brush border of our enterocytes, or enterocytes being a different way of referring to our gut cells, that is the epithelial cells that form the lining of our small intestine. If we take a look at the molecular arrangement of micelles, we find that it's actually quite similar to the phospholipid bilayer of our cell membranes. So the phospholipid bilayer of our cell membranes looks like this, where we have our phospholipids arranged to where the hydrophilic heads are on the outside of the cell membrane, and the tails, which are hydrophobic and do not like water, are facing each other. And in a micelle, of course, we don't have phospholipids, we just have our monoglycerides with fatty acid tails. We find the heads of our monoglyceride molecules facing the outside because they're hydrophilic, and then those fatty acid tails which are hydrophobic, point inward. So very similar to the phospholipid bilayer. Now, as soon as these micelles are in the proximity of these microvilli, they start to fall apart into their monoglycerides. I'll just put M for, or M and mono, I'll call it mono, and their fatty acids. And so these just readily diffuse into our cell, and they very quickly recombine to form new triglycerides inside of the cell. Now, these triglycerides are going to hook up with some cholesterol and some other proteins as well, for instance. And the Golgi body, or the Golgi apparatus, packages this all up and creates these new little structures that we will call chylomicrons. So the chylomicrons are being formed by these Golgi bodies after the triglycerides are reformed from the micelles, and the triglycerides combine with a few other products such as cholesterol and proteins and vitamins, etc. So these chylomicrons we see right here, these little packages formed by the Golgi body. And the chylomicrons are now going to be extruded from our cell, our epithelial cell, on the basolateral surface, such that they will be able to enter into the lacteals that are inside of our villi of our small intestine to become part of our lymph, fatty lymph. Now, why not enter the capillary or the capillaries? Well, the walls of the capillaries just cannot handle the size of these chylomicrons. They're just a tad bit too big, and consequently we see that the chylomicrons with their triglycerides and cholesterol, etc., are going to enter into the special lymphatic vessels we refer to as the lacteals. Now, this is okay because, as you remember, our lymph eventually gets returned to our blood, so these chylomicrons do eventually enter into the bloodstream. Once the chylomicrons are present in the blood, the cells that line our blood vessels, remember we call them endothelial cells, which are literally squamous cells, they themselves produce lipase, and the chylomicrons are small enough to where the lipases can easily break the triglycerides up into their end products of monoglycerides and fatty acid tails. And these very small molecules, since they're lipid soluble, can readily diffuse across the cell membrane and make it into the tissue cells so the tissue cells can use them. We also see that some of the monoglycerides and fatty acids can be stored in our adipocytes, or our hepatocytes, that is our liver cells, can use these monoglycerides and fatty acids to make lipoproteins to where we can carry and transport cholesterol in the blood. You might have heard of LDL and HDL. Let's do a quick review of what we've done so far. We've looked at how our monosaccharides and our amino acids and our fats are absorbed. So this is a nice chart. It's a little bit out of focus that summarizes this very nicely. It does not include the nucleic acids. We'll do these after this video. Glucose and galactose, which are monosaccharides, remember they depend on the pumping action of sodium to be able to be drawn in by diffusion on the apical surface of the columnar cell. So we call that secondary active transport. That's how the glucose and galactose make it into our epithelial cell, which is this yellow cell that we're showing here. And from there, these monosaccharides diffuse into our capillary. Fructose does not depend on secondary active transport. It can just diffuse into the epithelial cell of the villi and again diffuse into our capillaries. Amino acids also depend on secondary active transport and even some of our smaller peptides. Now, Fatty acids, if they're small, if they're short chain fatty acids, since they're lipid soluble, they can just depend on simple diffusion. Remember, the triglycerides, on the other hand, need to go through a more complex process to where they first become micelles with the help of bile salts and phospholipids in particular, and even lipases. These micelles can then fall apart into monoglycerides and fatty acids, which can very easily diffuse into our epithelial cell. There they recombine to form triglycerides, which get packaged up into chylomicrons, and they then diffuse into the lacteals. The lacteals, which are part of the lymphatic system, are going to eventually return the lymph back to the blood, and that's how these chylomicrons with their triglycerides ultimately end up in our bloodstream. Cholesterol is part of our bile. Cholesterol is mainly formed in the liver so that it can become part of the bile. But we also ingest cholesterol. And notice that we lose a lot of our cholesterol that we ingest actually in our feces. We only really absorb about 50% of the cholesterol that our liver synthesizes and that we ingest. Cholesterol is actually a very important molecule for our body. We have this very negative connotation when it comes to cholesterol because we've learned that cholesterol can cause major problems in our blood vessels. It can cause plaques such that we suffer from atherosclerosis. But let's not forget that we need cholesterol to build our cell membranes. Remember, the phospholipid bilayer has proteins as well as cholesterol. And cholesterol is a steroid molecule that forms the foundation for any other steroid molecules or many of our hormones, which are steroids. Think of testosterone and estrogen. Even vitamin D is a steroid and depends on cholesterol to be formed. Cholesterol is carried around, for instance, in the chylomicrons, and once we get to the blood vessels, then it can turn into what is called HDL and LDL. And these high levels of LDL are what can trigger atherosclerosis. I'm not going to go into any further detail. There is a little bit more of a description about atherosclerosis and cholesterol in your book, which you can read about. People who have unusually high cholesterol levels in their body can be put on drugs, and these drugs, as you might imagine, can impact the synthesis of bile at the level of the liver. So these drugs can impact the liver, or other drugs can impact the absorption rate of cholesterol. So this sums up our discussion of fat digestion and absorption. We still need to take a look at how we digest and absorb the nucleic acids and talk a little bit about some other nutrients, especially some of the electrolytes and vitamins.